Good day, grade 12 learners, and welcome to this video where we will be revising the first topic which you will be studying for your paper 1 of your NSC Life Sciences final examination. In this video, we will be revising the topic on human reproduction as well as reproduction in vertebrates using section B questions that I've compiled from previous trial examinations, specifically 2024's trial examinations. If we look at reproduction in vertebrates, according to your exam guidelines, the things that you should be studying is the diversity of reproductive strategies, the role of the following reproductive strategies in animals in maximizing reproductive success in different environments using relevant examples. You need to be able to differentiate between external and internal fertilization, oviparity, oviviviparity, and viviparity. The amniotic egg, that includes the diagram of the amniotic egg, the functions of the different parts in the amniotic egg, precocial and altricial development, the differences between the two, parental care. These were the things that we learned that you need to be able to understand within reproduction in vertebrates. The next type of reproduction that was looked at was human reproduction. According to the exam guidelines, we introduced the topic. Then we looked at the structure of the male reproductive system. You need to know the structure of the male reproductive system using a diagram. The functions of the testes, the epididymis, the vas deferens, seminal vesicle, prostate gland, corpus gland, penis, and urethra. Then the structure of the female reproductive system, you need to know that as well. The structure of the female reproductive system using a diagram, functions of the ovary, fallopian tubes, uterus, lined by the endometrium, the cervix, the vagina with its external opening, and the vulva. Then the structure of the ovary using a diagram showing the primary follicles, the Graafian follicle, and the corpus luteum. Then we also looked at puberty, the main changes that occur in male characteristics during puberty under the influence of testosterone. You need to be able to name a few of those changes. The main changes that occur in female characteristics during puberty under the influence of estrogen. You need to list a few of those changes. Then the process of gametogenesis, the formation of gametes by meiosis, the male gametes formed by spermatogenesis, female gametes formed by oogenesis. The process of spermatogenesis, very, very important. You need to be able to describe this entire process. If you are asked to describe the entire process of spermatogenesis, the process of gamete formation in males, please make sure you are able to describe it like this from the exam guidelines. Under the influence of testosterone, diploid cells in the seminiferous tubules of the testes undergo meiosis to form haploid sperm cells. Then you need to be able to draw and label the structure of a sperm cell, the functions of the parts of a sperm cell, which includes the acrosome, the head, the haploid nucleus, the middle portion with the neck that also comprises of the mitochondria, and the tail. The functions of these parts you also need to know. Then the process of gametogenesis in females is referred to as oogenesis. Please study the process of oogenesis from the exam guidelines, so that you are awarded all your marks for it. Diploid cells in the ovary undergoes mitosis to form numerous follicles. At the onset of puberty and under the influence of FSH, one cell inside a follicle enlarges and undergoes meiosis. Of the 4 cells that are produced, only one survives to form mature haploid ovum. This occurs in a monthly cycle. Then the structure of an ovum or egg cell, you need to be able to draw and label the structure of an ovum or egg cell. The functions of the different parts of the ovum, the jelly layer, the haploid nucleus, and the cytoplasm. The functions of those parts you need to know as well. Then we looked at the menstrual cycle. The menstrual cycle included the uterine and the ovarian cycles. We looked at the events in the ovarian cycle, which included the development of the Graafian follicle, the process of ovulation, the formation of the corpus luteum. Then the events in the uterine cycle, the changes that takes place in the thickness of the endometrium, as well as the process of menstruation. You would have also discussed hormonal control of the menstrual cycle, the ovarian and uterine cycles with reference to the action of FSH, estrogen, LH and progesterone. Very, very important. Negative feedback mechanism involving FSH and progesterone in controlling the production of ova. This is also extremely important. Then the next aspect that was touched on was fertilization and the development of the zygote to the blastocyst, definition of copulation and fertilization, defining the terms, the process of fertilization, what happens in fertilization, the development of the zygote to the embryo, where it first goes into the morula, then it goes to the blastocyst, and then finally it goes to the fetus. Then the last aspect of this topic was implantation, gestation, and the role of the placenta, definition of implantation, the role of estrogen and progesterone in maintaining pregnancy, the structure of the developing fetus in the uterus using a diagram, the functions of the following parts, the chorion and the chorionic villi, the amnion, the amniotic cavity, and the amniotic fluid, the umbilical cord, including the umbilical artery and the umbilical vein, and then lastly the placenta. Please make sure that when you are studying for this topic, you are going through everything in the exam guidelines and you have covered all aspects as stipulated in the exam guidelines, according to what I have just illustrated to you now. The first question that we will look at is taken from a question 2.1. We will specifically focus on section B, questions on human reproduction. It says, read the extract below. In South Africa, 84,000 preterm births occur annually. Preterm babies are those who are born alive before the full 40 weeks of pregnancy. 10% of these babies born are likely to die or experience acute respiratory, neurological, and eye problems compared to full-term babies due to being underdeveloped. The bio bag in the diagram below aims to create an artificial uterus for the developing fetus to continue its development in. Studies have shown that this bio bag can be used to provide a lamb with all the necessary conditions to gestate to full term. If we have a look at the actual structure given to us in the diagram, we have a thick plastic bag, we have the fetus, the umbilical attachment, inlet for purified amniotic fluid, oxygenator, and the intravenous bag where the hormones, anticoagulant, and nutrients are going to be in. The first question, 2.1.1, says calculate the number of premature babies born in South Africa that are likely to die or experience acute respiratory, neurological, and eye problems. For 3 marks calculation, we need to show the calculations. So if we look in the first paragraph, you can see in South Africa, 84,000 preterm babies, preterm births occur annually. Preterm babies are those who are born alive before the full 40 weeks of pregnancy. 10% of these babies born are likely to die or experience acute respiratory, neurological, and eye problems compared to the full-term babies. This sentence coincides with the question. So we want the number of premature babies that are likely to die or experience acute respiratory, neurological, and eye problems. So only 10% of the babies that are born are going to be experiencing those problems. But how many preterm births are there? There are 84,000. Therefore, to answer 2.1.1, we are taking 84,000 and we are simply calculating 10% of it. 10% is 10 out of 100, multiplying by 84,000. type that in the calculator, it will give you 8,400 babies. 3 marks for 2.1.1. The next question, 2.1.2, identify one component of the biobag and explain how it would act as the amnion for 2 marks. Identify one component of the biobag. So in each of these, in A and in B, we're going to only identify one component of the biobag that serves as the amnion. And then we're going to explain how it would act as the amnion. So we're going to look in the biobag. We first ask ourselves, what does the amnion actually do? And what is the actual function of the amnion? And which structure do we have in this diagram that's going to perform that similar function? So if we look at A, we are looking at the amnion. If we look at the sac here on the outside, we have a thick plastic bag. That thick plastic bag is serving as the amnion. Why is it serving as the amnion? It encloses and protects the developing fetus until delivery. And that's generally the purpose of the amnion. So the thick plastic bag is enclosing and protecting the developing fetus until delivery. And it serves as the amnion. Then B, the placenta. Now we look which section on this diagram is showing us the placenta. You also have to think about the function of the placenta in order to get the correct answer. So if we look at this oxygenator here, the oxygenator is going to be attached to the growing fetus. And it's going to allow certain things to pass through. All those functions that we're going to have of the placenta, we think which one of it is best going to coincide with the oxygenator. What would the oxygenator most likely do? It's going to carry gases. It's going to allow for gaseous exchange. And that is the function that coincides with the function of the placenta. So therefore we say the oxygenator acts as the placenta because it allows for gaseous exchange. The next question, 2.1.3 says, describe how the biobag could eliminate risks of underdeveloped organs in preterm babies for 3 marks. How would the biobag eliminate risks of underdeveloped organs in the preterm babies? So you first have to ask yourself, are babies able to fully grow and develop in this biobag? Yes, they are. Because it says that it's going to be used to provide a lamb with all the necessary conditions to gestate them to full term. Therefore, to answer the question, we can say that the developing fetus or organs is allowed to continue developing, giving the organs sufficient time. So the organs have sufficient time to develop. It's normal development, so it's going to be given time for the organs to develop so that the fetus could develop fully. And once the fetus is developed fully, scientists are then able to determine when the gestation period is going to be over and when it will then be time to deliver the actual baby. So 3 marks for 2.1.3. 2.1.4, you are listing 2 ethical issues that would need to be considered before the introduction of this reproductive technology on human trials for 2 marks. What are they actually asking here? 2 ethical issues that should be considered before this process actually takes place. What are you actually going to need to consider? Firstly, human fetuses could possibly be lost or destroyed. So in this process, human fetuses could possibly be lost or destroyed, because the fetus is not going to be growing in the normal womb like it normally does. Acquiring consent from regulatory bodies, parents, you need to acquire consent from certain people. And last and most importantly, fully informing donor parents about the risk. The donor parents need to be informed about the risks it's involved in this process before you're actually going to undergo it. So any 2 of these points for 2.1.4, you would have awarded 2 marks because 2 ethical issues. Last one that you could possibly have said was, it's contrary to religious observations. So some religious beliefs will not agree to this actual process, and therefore it could be a disadvantage to those who actually want to start it. 2.2, read the extract below. The red kangaroo is the largest of all kangaroo species and is endemic to Australia. During copulation, the male releases semen inside of the female's body and gestation takes 33 days until birth. The blind, naked and stump-legged baby kangaroo must crawl its way up the mother's fur to a pouch where it attaches onto a tree, then provides nourishment and remains protected there for the next 190 days until it becomes a joe. At 235 days, the joey is ready to leave the mother's pouch full time. So here you have the pouch containing a joey. And here the baby is coming out. So 2.2.1, identify the type of fertilization that takes place in the red kangaroo for 1 mark. There are 2 types of fertilization. It is either internal or external fertilization. And you will notice here it says during copulation the male releases semen inside the female's body. That is an indication that we are looking at internal fertilization. Therefore 2.2.1 internal fertilization. 2.2.2 asks you to give one reason for your answer in question 2.2.1. So the male releases semen inside of the female's body. And that is why we can say it is internal fertilization. Question 2.2.3 says identify the reproductive strategy used by the red kangaroo where its young are born alive. There are one of 3 reproductive strategies. It's either oviparity, viviparity, or ovoviviparity. So we are going to look how is this baby kangaroo actually going to be growing. And where is the growth going to take place? So we see, if we read the paragraph, it says the blind, naked and stump-legged baby kangaroo must crawl its way up the mother's fur to a pouch where it attaches onto a teat that provides nourishment and remains protected there. If it's going to gain its nourishment from the inside of this kangaroo, it's going to be evident that it was growing inside this kangaroo's body. Therefore, it is going to be viviparity. So 2.2.3, viviparity. 2.2.4, discuss one parental strategy the red kangaroo displays that ensures a high survival rate of its young. One parental strategy that ensures a high survival rate. So firstly, for 2.2.4, the baby kangaroo is able to develop within its mother's pouch for an extended period of time till 235 days. And secondly, this gives greater protection against environmental threats. So because the baby kangaroo is able to develop within its mother's pouch for an extended period of time, and this gives greater protection against environmental threats, it's a type of parental strategy that ensured a high survival rate of its young. 2.2.5, while you describe the red kangaroo as altricial. So, why do we describe it as altricial development? If we look in the paragraph, it clearly states the blind, naked and stump-legged baby kangaroo must crawl its way up the mother's fur. Blind, naked and stumped. That is a clear indication. The baby kangaroo is blind, the baby kangaroo is naked, and therefore it relies on the parent for nutrition also, because that provides nourishment onto a tree that provides nourishment. That's an indication that it's dependent on the parent for nourishment. The baby kangaroo is blind, naked and relies on the parent for nutrition. That is why we can say it is going to be altricial development for 2.2.5. The next question is taken from 3.3 that says the graph below shows the changes in female hormones during the menstrual cycle. So you have estrogen, progesterone and luteinizing hormone over a 28-day period. If we have a look on the x-axis, we have the days. On the y-axis, we have our hormone concentration. And then we have keys here for estrogen, progesterone, as well as luteinizing hormone. Question 3.3.1 says identify the structures that secrete estrogen from days 1 to 13. So we look at days 1 to 13. Estrogen, so we're looking at the solid line, day 1 to 13. So identify the structures that's going to be releasing this estrogen here from days 1 to 13. Which structure releases estrogen? You are going to have the ovaries releasing estrogen. And specifically inside the ovaries, the Graafian follicle is going to release estrogen. So whether you say the ovaries or whether you say Graafian follicle, it will be accepted. Then B, the progesterone from day 13 to 20. We are now looking at progesterone levels. So this thick dotted line from day 13 all the way to day 20. What is releasing this high levels of progesterone here? What structure starts to release progesterone? The corpus luteum that is going to be located inside the ovary. The next question, 3.3.2, on which day did ovulation take place for 1 mark? If we look in the diagram on the graph, we can see that luteinizing hormone, the dotted line, spikes up here on day 14. So therefore ovulation took place on day 14, 3.3.2. For 3.3.3, give one reason for your answer. So we just stated the reason now. The luteinizing hormone levels have spiked up or have peaked around day 14. Question 3.3.4, explain why progesterone levels remain high after day 21 for 2 marks. Now we look at day 21. Here we have day 21. We're looking at our progesterone levels, so it is this levels here that we are referring to. Explain why progesterone levels remain high after day 21 for 2 marks. So why would progesterone levels remain high? Generally, your progesterone levels are remaining high because implantation or fertilization has occurred. If fertilization has occurred, it therefore means that the corpus luteum does not degenerate. It continues to, it remains in size, and therefore it continues to secrete the hormone progesterone. 3.3.5, why is it not possible for a female to develop another Graafian follicle during pregnancy? There's 4 marks for this question. Please take note of that. This question is something that could be examined. Please make sure you understand what this is about. Why is it not possible for a female to develop another Graafian follicle during pregnancy? Emphasis on during pregnancy. If pregnancy has occurred, then firstly the corpus luteum will remain in size. The endometrium lining is going to remain thick, and progesterone levels will remain high. If your progesterone levels remain high, it's going to inhibit the pituitary gland from releasing follicle-stimulating hormone. If follicle-stimulating hormone is not released, then a new primary follicle cannot be released and it cannot grow into a Graafian follicle. So to answer 3.3.5, high levels of progesterone will inhibit the pituitary gland from secreting FSH, follicle stimulating hormone. No follicles will be stimulated to develop. So the FSH will not stimulate the release of the primary follicle to then grow into that Graafian follicle. Please make sure you are able to understand a question like this. It is testing your knowledge on negative feedback between progesterone as well as FSH. Question 2.5, the diagram below shows the parts of the male reproductive system. So you have the male reproductive system in this diagram with letters A and letter B. 2.5.1 asks you to identify part A. What is part A? The structure here. What are we referring to? We are referring to the seminal vesicle. So 2.5.1, we are referring to the seminal vesicle. Then 2.5.2, the prostate cancer affects men and the risk increases as men get older, which may lead to surgical removal of part B. Explain how the removal of part B may affect male fertility for 3 marks. First, you're going to look at part B and you see that part B is referring to the prostate gland. Now you ask yourself, what is the function of the prostate gland? So the prostate gland normally releases an alkaline medium that's going to protect you from the low pH conditions that's found in the urethra. If the prostate gland cannot release that alkaline medium, you won't be protected from the low pH conditions at the urethra as well as in the vagina. So sperm cells will die out and therefore it will decrease your fertility. So for 2.5.2, the alkaline fluid that's normally released by the prostate gland will not be secreted. It will not be secreted, therefore it cannot neutralize the acid in the urethra, and this acid is specifically referring to that low pH conditions. Sperm cells will denature and die out, and it will decrease your male fertility. 3 marks for 2.5.2. 2.5.3 says describe the process of spermatogenesis for 4 marks. This is exactly how they could ask you the question. You study it from your exam guidelines, which states that under the influence of testosterone, your diploid cells in the seminiferous tubules of the testes are going to undergo meiosis to produce haploid sperm cells. 4 marks for 2.5.3. Please make sure you are able to describe the process of oogenesis as well, which is the process of gametogenesis in females. 2.6 says scientists at Harvard University investigated to determine which type of underwear leads to low sperm count. They asked 656 men to provide a semen sample and answer a questionnaire about what type of underwear they wore most often in the previous 3 months. In the results, they found that 75% of men who wore loose-fitting boxer shorts had a higher sperm count, while 25% of men who wore tight underwear had a low sperm count. 2.6.1 name the part of the male reproductive system where sperm cells are stored. So sperm cells are generally going to be stored inside the epididymis. 2.6.2 state one way in which the results of the investigation can be considered reliable. When we talk about improving reliability, what do we normally do? We repeat our investigation, we increase our sample size. Now, if you looked in the paragraph, they see there are 656 men to provide a semen sample. But they didn't only ask one man or 2 men. They chose 656. So what did they do? They increased the sample size. Therefore, we say 656 men participated in the survey. That is what they did. Next question, 2.6.3. Explain why wearing loose-fitting underwear will increase male fertility for 3 marks. So firstly, wearing loose-fitting underwear is going to allow the testes to hang further away from the body. If the testes hangs further away from the body, what does that mean? The temperature of the testes will be 2 to 3 degrees Celsius lower than the body temperature. That is going to allow for optimum sperm production, where your sperm cells are now going to be healthy. 2.6.4 except for movement. Describe one way in which sperm cell is structurally adapted to fertilize the ovum for 2 marks. So besides movement, how is a sperm cell adapted to fertilize the ovum? Now you need to know the exact structure of the sperm cell. It has the head comprising of the acrosome and the nucleus. It has the body with the mitochondria, then it has the tail. Which part is going to allow it to fertilize the ovum? So the head is going to comprise of an acrosome. The acrosome consists of lytic enzymes, and those enzymes are going to help to penetrate the outer membrane of the ovum. So what is the one way? The head is going to comprise of the acrosome, which contains lytic enzymes that helps to penetrate the outer membrane of the ovum. 2 marks for 2.6.4. The next question, 3.3, says the diagram below represents a developing fetus in the uterus. So we have our developing fetus, we have part D, part A, part B, and part C. 3.3.1 says identify label B for 1 mark. So we're only identifying label B. What is this referring to? It's referring to the chorion. 3.3.2, name 2 functions of A. So 2 functions of A. It's this fluid that we have here on the inside, the amniotic fluid. There are numerous functions of the amniotic fluid. It acts as a shock absorber. It acts as a temperature regulator. It prevents dehydration, and it allows a medium for the fetus to move in. Question 3.3.3 says describe how structure C is formed for 3 marks. Structure C, this is what we are referring to. How is the structure formed here? So what do we have here? We have the placenta. We then have the chorion. The chorion formed the chorionic villi. So we're answering that. We are saying that the chorion forms the chorionic villi and attaches itself to the endometrium. That's simply what we are describing. So the chorion formed the chorionic villi, which attached itself to the endometrium, for 3 marks. 3.3.4, explain the endocrine role of part C during gestation, for 3 marks. Whenever you see the term endocrine role, then you need to think they're talking about a hormone. They are referring to some sort of hormone when they're talking about endocrine role of part C during gestation. What is gestation? It's that period between implantation and birth. Part C, this is part C. Explain the endocrine role of part C during gestation. Endocrine role, what hormone is the only hormone they could be referring to that's released here? What part is that? The placenta. Remember that once you are going to have the fetus growing and developed and you have the placenta, that placenta is now going to start releasing progesterone. So it's going to secrete progesterone. And what is the progesterone going to do? It's going to further increase the thickening of the endometrium. And when it increases the thickness of the endometrium is going to become more vascular and more glandular. That's for 3.3.4, 3 marks for the question. 3.3.5, tabulate 2 differences between the composition of blood found in the blood vessels in part D for 5 marks. Blood vessels in part D. What is part D pointing to? It's pointing to the umbilical cord. What are the blood vessels in the umbilical cord? You are going to have the umbilical artery and the umbilical vein. So we are tabulating differences between the umbilical artery and the umbilical vein. So for 3.3.5, you draw your table so that you get 1 mark for your table. Then you are going to have the umbilical artery and the umbilical vein. The umbilical artery carries deoxygenated blood from the fetus to the placenta, while the umbilical vein carries oxygenated blood from the placenta to the fetus. That's one difference. Another difference, the umbilical artery carries toxic substances from the fetus to the placenta, while the umbilical vein carries nutrients from the placenta to the fetus. 2 differences that you would have stated, 1 mark per statement and 1 mark for the table gives you 5 marks for 3.3.5. The next question, 3.4, says read the extract below. Shark reproduction. In hammerhead, lemon and mako sharks, the embryo develops inside of the womb and will have a placenta and live birth. Each shark has an umbilical cord that is located between the pectoral fins. The bamboo sharks and epaulet sharks lay eggs in a deposit of water. Once the eggs are laid, the female leaves without the eggs being protected. The female can lay 200 eggs. In great white sharks, nurse sharks, and thresher sharks, the egg hatches inside the womb and the shark has a live birth. If we look at question 3.4.1, name one ovoviviparous shark. What does ovoviviparous shark mean? That this shark is going to be growing inside the mother's womb, but it's going to have an egg as well. If we look at this last statement, in great white sharks, nurse sharks, and thresher sharks, the egg hatches inside of the womb and the shark has a live birth. That coincides with 3.4.1. Therefore, we can say great white shark, nurse shark, or thresher shark. One of them would have given us 1 mark. 3.4.2, explain why bamboo sharks and epaulet sharks release large numbers of eggs. Now this is quite obvious. You generally release large numbers of eggs to increase your chances of fertilization. Since it is external fertilization, because when external fertilization occurs, it's easy for the eggs to go away and the eggs may be lost. So as I've mentioned, if external fertilization is going to occur, it's easy for the eggs to be lost. So you want to release large numbers of eggs to increase the chances of fertilization, since it's external fertilization and the eggs or ovums may be lost. The next question, 3.4.3, says describe the differences in nutrition of the embryos of the lemon and bamboo sharks for 3 marks. Differences in the nutrition of the embryos. How do the embryos receive nutrition of the lemon and bamboo sharks? So if you go to the lemon sharks here, it says each shark has an umbilical cord that's located between the pectoral fins. The bamboo sharks and epaulet sharks lay eggs in a deposit of water. Once the eggs are laid, the female leaves without the egg being protected. So you can see that the lemon shark develops inside the womb. But the bamboo sharks and epaulet sharks lay eggs in a deposit of water. That means that the lemon shark is going to obtain his nutrition from the womb through the placenta. The bamboo shark will receive their nutrients from the egg, and in the egg you normally get your nutrients from the yolk that's found inside the amniotic egg. So therefore the lemon shark has a womb and the embryo is fed by the placenta or the umbilical cord, while the bamboo shark embryo are fed by the yolk in the egg. That's for 3.4.3. The next question is a 2.1. It focuses on the menstrual cycle. Very important. Please make sure you are able to understand the menstrual cycle. It says study the diagrams and graphs below which show the hormonal regulation of the female reproductive cycle. So you have your different parts here, the pituitary, hormone levels, growth of the follicle, ovarian hormone levels, thickness of the uterine lining or endometrium, and hormonal regulation of the female reproductive cycle. Question 2.1.1 says give the number and name of the hormone that stimulates the development of the follicle for 2 marks. The number and name of the hormone that stimulates the development of this follicle here is going to be number 1. Number 1 is referring to follicle stimulating hormone. B says maintains the endometrium between day 7 and 14. What is maintaining the endometrium here? These levels, which is what number? Number 4, and it's going to be the hormone estrogen. 2.1.2, explain the function of the corpus luteum for 2 marks. What does the corpus luteum do? The corpus luteum is going to secrete progesterone, and progesterone is going to maintain the endometrium. Then B, describe how the corpus luteum is formed. So how does this corpus luteum actually form? If we go back, you can see here that this high amount of luteinizing hormone triggered that process of ovulation. What released luteinizing hormone? The pituitary gland. So the pituitary gland released luteinizing hormone, which triggered that process of ovulation. And when the remains of that Graafian follicle then form the corpus luteum. So for 2.1.2b, the pituitary gland or the hypophysis secretes luteinizing hormone, which stimulates the conversion of that ruptured Graafian follicle into a corpus luteum. 3 marks for 2.1.2b. 2.1.3, give 2 visible reasons using the information from the diagrams and graphs to explain why the woman is not pregnant for 2 marks. So the woman is not pregnant. Firstly, you're going to be looking at your corpus luteum. You can see there the corpus luteum is shrinking and disappearing. Secondly, you look at your levels of progesterone. Your levels of progesterone are dropping towards the end of the menstrual cycle. So there you have 2 reasons. The corpus luteum is getting smaller. Progesterone levels are dropping. 2 visible reasons that we took from the diagram. You could also say that the FSH levels are increasing on days 27 to 28. If you look around day 28 and you look here, the FSH levels, it's starting to go up again. And that indicates to you that the woman is not pregnant, because if she was pregnant, you would have had high levels of progesterone, and that high levels of progesterone would have inhibited the pituitary gland from releasing FSH, and therefore the FSH hormones would have remained low. That brings us to the end of this video, where we revised human reproduction as well as reproduction in vertebrates using section B questions from previous exam papers. Please make sure that when you are studying for paper 1, you are studying the correct topics, namely reproduction in vertebrates and human reproduction, responding to the environment in humans, which comprise of the nervous system, responding to the environment in plants, which comprise of plant hormones, and then lastly the endocrine system and homeostasis. In this video, we completed reproduction in vertebrates and human reproduction using section B questions from previous exam question papers. In the next video, we will focus on responding to the environments in humans, where we will revise the human nervous system. Thank you very much for taking your time to watch this video. I hope to have you all join me in the next video, which will be revising the human nervous system.